Hello everyone, myself Dr. Amanpreet Kaur Kalsi. In this lecture, we'll take up another topic from Developmental Biology, Early Development in Frog. The topics we'll cover in this lecture are Fertilization and Cortical rotation, Cleavage, which is the process of blastulation, Midblastula transition, Fate maps in frog, and Gastrulation of frog. Let's first see an overview of development of frog. Germ cells are produced in adult frog by the process of gametogenesis. These germ cells undergo fertilization to give rise to zygote. As soon as the zygote is formed, it undergoes cleavage process where morula and blastula are formed. Blastula then enters the process of gastrulation where the three germ layers are formed: ectoderm, mesoderm and endoderm. Up till the formation of three germ layers, the process is called gastrulation, and this is the early development phase. Then the embryo enters the process of organogenesis and then forms the adult, going through some larval stages. In this lecture, we will cover early development of frog up till the process of gastrulation, that means formation of three germ layers. Let's start with fertilization and cortical rotation in frogs. This figure shows schematic cross-section of a frog egg midway through first cleavage cycle. Fertilization in frogs occur anywhere in the animal hemisphere. The point of sperm entry is very crucial as it determines the orientation of dorsal ventral axis. The side from where sperm enters becomes the dorsal side of the embryo, and the side 180 degree opposite the sperm entry side becomes the ventral side of the embryo. As sperm enters, the sperm centrioles organizes the microtubules of egg cytoplasm into parallel arrays as are seen in this figure. At the same time, a layer of cytoplasm at the cortex becomes separated from the internal yolk-laden cytoplasm, and this layer is called cortical cytoplasm. The microtubular arrays then causes this cortical cytoplasm to rotate by an angle of 30 degree with respect to inner cytoplasm. This exposes a band of inner cytoplasm which is called gray crescent in the marginal region. This is the region where gastrulation starts later on. As the cortical rotation is over, the microtubular arrays also disappear. So immediately after fertilization, the zygote enters the process of cleavage. The kind of cleavage depends upon the type of egg frogs have. Frogs have meso-lecithal eggs. Meso means moderate, lecithal means yolk. So they have moderate to large amount of yolk which is largely concentrated in the vegetal hemisphere. This yolk interferes in cleavage process. So cleavage in the vegetal region is delayed compared to cleavages in the animal region, and that is why the cleavage in frog is unequal, radial, holoblastic. Unequal because the cleavage in animal and vegetal region are unequal because of differential presence of yolk in these two regions. Let's see the process of cleavage. The first cell division is meridional, starts at the animal pole, slowly extending towards the vegetal region, crossing through the center. The second division is also meridional and right angle to first, but because of large amount of yolk in vegetal region, even before the first cleavage completely reaches the vegetal end, the second cleavage already starts at the animal pole. The third cleavage is supposed to be equatorial and perpendicular to the first two cleavages, but again because of large amount of yolk in the vegetal region, it gets displaced towards the animal pole. So here is where third cleavage occurs. As a result of this, smaller sized blastomeres are formed at the animal pole called micromeres, and larger sized blastomeres are formed at the macromeres, which are also yolk laden, and these are called macromeres. Despite this unequal sizes of blastomeres, they continue to divide at the same rate until 12th cell cycle, with only a small delay at the vegetal cleavages. This gives rise to numerous small micromeres at the animal hemisphere and relatively larger and fewer yolk-laden macromeres at the vegetal region. The embryo from 16 to 64 cell stage is called morula, plural is morulae, which means mulberry, and it is named so because its shape vaguely resembles mulberry. As cleavage proceeds, the 128 cell stage is reached and blastocele becomes apparent, and now the embryo is called blastula. The formation of blastocele actually can be traced back to the very first cleavage. As the very first cleavage occurs, the cleavage furrow widens in the animal hemisphere and gives rise to a small intercellular cavity. This cavity then seals off from the outside by tight intercellular junctions. Gradually, the cavity expands, subsequently giving rise to blastocele by blastula stage. The tight intercellular junctions that we are talking about are maintained by cadherin molecules, which are calcium-dependent cell adhesion molecules. The cadherin molecules which are present here are mostly EP cadherins because they resemble both epithelial cadherins and placental cadherins. So they are named EP cadherins. This shows an enlarged view of a cadherin molecule. The first domain contains binding site for other cadherin molecules. They have calcium binding sites, and then they have sites for binding 3 catenin molecules, which then bind to the actin molecules in the cell cytoplasm. So this is how the tight intercellular junctions are maintained. The blastocele actually plays 2 major functions. Firstly, it allows cell migration during gastrulation, and second, it prevents the cells below it to interact prematurely with the cells above it. The cells above and below the blastocele have different fates. They are destined to form different paths. So if they interact prematurely, it results in abnormality. The process of cleavage is regulated by certain factors called mitosis promoting factors, which are composed of 2 components, cyclin B and cyclin dependent kinases. Up till the first 12 divisions, there are no gaps in cell divisions. So cleavage continue to occur one after the other until 12th cell cycle. These cell cycles have mitosis and synthesis phases only. There is no growth phase, no G1 and G2 phases. Now because there is no growth phase, so the size of embryo remains the same during entire cleavage process. Only the nuclear content keeps on increasing. As a result, the cytoplasmic to nuclear ratio decreases, or alternately we can say that nuclear to cytoplasmic ratio increases. But at 12th cell cycle, brought about by a change in this chromatin to cytoplasmic ratio, there occurs demethylation in promoter region of certain genes. As the promoter regions become demethylated, now some transcription factors can bind to these promoters. These transcription factors are largely produced by the cells of vegetal hemisphere. As the transcription factors bind, the transcription is initiated in these genes. So different genes get transcribed in different cells. Thus, the blastomeres acquire the capacity to become motile. This dramatic change is called midblastula transition, where the embryo is preparing to move from blastulation to gastrulation process. Why are they moving? Because they have different fates. They are destined to form different cells, different organs of the body. They have to be present in their final position than when they are now. Now, which cell is supposed to form which final part or ultimately which region of the body? This we come to know with the help of fate maps. Fate maps are diagrams which map larval or adult structures onto region of embryo from which it arose. Fate maps provide information on which portion of the embryo normally becomes which larval or adult structures. This shows fate map of Xenopus frog. The first figure shows a dorsal view, that means from the side, which in future will become the back of the animal. And the other two figures show the side view. The first one is the exterior side view and the second is cross section showing the interior side view. Now follow the color codings. The blue color shows the ectoderms. That means the cells in these regions will form the ectoderms or ectodermal derivatives in future. The yellow color shows the endoderms. So these cells will form endodermal regions in future. And the red and dark red color are the mesodermal regions. So as we can see, the prospective ectoderm and endodermal cells are largely present on the surface of the embryo, while the mesodermal cells are lined in the equatorial zone in the marginal region, mainly in the interior of the embryo. Now let's come to the process of gastrulation, where these cells which are present at different places will ultimately come to lie at their final positions where they're supposed to be. So gastrulation is the process of highly coordinated cell and tissue movements, whereby cells of blastula are dramatically rearranged to give new positions and multi-layered body plan of organism is established. The three germ layers, outer ectoderm, inner endoderm and interstitial mesoderm are first produced during the process of gastrulation. Now since gastrulation involves movement of cells and tissues, that is why the process of midblastula transition occurred, where the cells of embryo acquired the capacity to move. Now gastrulation is a combination of some basic kinds of movements. Let us see those movements once. The first is invagination, which is infolding of cell sheet into the embryo, as is shown here by the yellow colored sheet of cells which is infolding or invaginating into the embryo. This kind of movement is seen in sea urchin endodermal cells. Next is involution, which is inturning of cell sheet over the basal surface of an outer layer, as is shown here by the yellow colored sheet of cells which is inturning or involuting over the blue colored sheet of cells. This kind of movement is seen in amphibian mesodermal cells. Next is ingression, which is migration of individual cells into the embryo. Here, the yellow colored cells are ingressing individually into the embryo. This kind of movement is seen in sea urchin mesoderms and Drosophila neuroblasts. Then is delamination, which is splitting of one sheet into two sheets, as is shown here, the blue colored sheet of cells. This kind of movement is seen in mammalian and bird hypoblast formation. And the last one is epiboly, which is expansion of one sheet of cells over other. Here, epiboly is shown by the blue colored sheet of cells which are covering the yellow colored sheet of cells on the outside. This kind of movement is seen in ectoderm formation in amphibians, sea urchins and tunicates. Once we know these basic movements, now let's move to the process of gastrulation in frog. In frogs, the gastrulation is brought about by some steps which include vegetal rotation. That means the cells in the vegetal region undergo rearrangement. The process is called vegetal rotation. Then there undergoes invagination of bottle cells. Some bottle cells are formed, as we'll see later on, and these move by the process of invagination. Then occurs involution at blastopore lip, convergent extension of dorsal mesoderm, and epiboly of prospective ectoderm. Let's see how it occurs. Now this figure shows early movements of frog gastrulation showing vegetal rotation. The color coding is same and is maintained in all the figures. The blue one shows prospective ectoderm, the yellow one shows prospective endoderm, and the red shows prospective mesoderm. Now as we talked earlier, the grey crescent region, which is the area 180 degree opposite the sperm entry site, is the region where gastrulation is initiated. So this is the site of blastopore formation, where gastrulation will be initiated and blastopore will be formed, as we will see. Now in this marginal zone, the prospective mesoderm cells are differentiated into cauda mesoderm cells, which in future will form the notochord, and head mesoderm cells, which will form the head region of the adult. These two group of cells forms the IMZ, that means involuting marginal zone. So these are the cells in the marginal zone which will undergo involution, as we will see later. Also, there are cells which will form the pharynx in future, the pharyngeal endoderm cells. Now what happens? The deep cells in the vegetal endoderm region undergo rearrangement. As a result of this cellular rearrangement, the pharyngeal endoderm, the one shown in orange color, comes to lie on side of prospective ectoderm and above the head mesoderm. The deep cells then progresses further into the blastocele just below the prospective ectoderm. Along with these, the pharyngeal endoderm cells and the involuting marginal zone cells are also drawn inside the embryo passively. So the actively moving cells are those of the deep layers of vegetal endoderm, along with which the involuting marginal zone cells move passively. Also, as these cells move inside, a slit opens on the surface which is called dorsal blastopore lip. As the cells on the surface start involution, a group of cells which are being dragged towards the inside while still maintaining contact with the surface appear, and because they are elongated, because one end of them is being dragged inside and the other end is still maintaining contact with the surface, they acquire the shape of a bottle. So these are called bottle cells. Ultimately, as the cells crawl towards the animal pole, these bottle cells and IMZ cells are also dragged towards the inside. This figure shows cell movement during frog gastrulation, showing invagination of bottle cells during early gastrulation. This is a meridional section showing three-dimensional view. The same color coding is followed. The blue are the ectodermal derivatives, the yellow are endodermal derivatives. AP refers to animal pole, opposite is the vegetal pole. This is the blastocele. These are the superficial cells and these are the deep cells. So these are the deep cells in the vegetal region which where vegetal rotation starts and involution is initiated. As these cells undergo involution and the marginal mesodermal cells also are dragged with them, a slit appears on the surface which is called dorsal blastopore lip. As these cells move further and further into the embryo, the blastocele is displaced from the animal pole towards the vegetal pole. Along with this, the animal pole is also displaced. The mesodermal cells are moving further and further inside the embryo by the process of convergent extension. At the surface, they are converging and then they are extending in the inside. So, convergent extension of mesoderm. As these cells are moving inside the mesodermal and endodermal cells, another new cavity appears which is called archenteron. This in future forms the foregut of the animal. As the dorsal mesodermal cells are involuting, another group of cells appear on the ventral side. These are also bottle cells which undergo involution to ultimately give rise to ventral mesoderm cells. As more and more cells involute the endodermal and mesodermal cells, the blastocele is further displaced and reduced. Ultimately, the blastocele is completely obliterated. Archenteron becomes larger in size, still maintaining contact with the outside surface, and this pore in future forms the anus of the animal. Also, the dorsal lip gradually extends on the sides, forming lateral blastopore lip, and on the ventral side also forming ventral blastopore lip. The endodermal cells which are then visible through this blastopore are called yolk plug. The dorsal mesodermal cells which involute forms future notochord. The cavity is the archenteron. At the same time, when the mesodermal and endodermal cells are involuting, the ectoderm covers the entire surface of the embryo by the process of epiboly. So as we can see, the blue colored cells have completely covered the outer surface of the embryo. Just the yolk plug area is left. Also, from the ventral side, mesodermal cells have involuted, forming the ventral mesoderm, as is shown in this figure. So this is a final image of gastrulation where the ectoderm has completely covered the outer surface of the embryo. All the endodermal cells, the yellow colored ones, have been internalized, and mesoderm has come to lie in between the ectoderm and endoderm. Up till here is the process of gastrulation. This figure shows all the 6 images together, the gastrulation process. Now during each stage, how it looks from the surface can also be seen here. Initially, there is just a small dorsal lip which gradually extends on the sides, then forms lateral lips, also forms ventral lip, giving more surface for the cells to involute and ultimately giving rise to blastopore with the endodermal cells which are visible, which are then called yolk plug. Gradually, the ectoderm completely seals this also, and the yolk plug is then not visible. This is all about gastrulation in frog. Let's see summary. So initially, fertilization occurred in the animal region, giving rise to zygote. As it is formed, it enters the process of cleavage, forming morula and then blastula. Blastula then enters the process of gastrulation, where 3 germ layers, ectoderm, mesoderm and endoderm, are defined. Then during later developmental stages, these 3 layers give rise to rest of the parts of body. The ectoderm forms skin epidermis, sense organs and central nervous system. Mesoderm forms bones, muscles, excretory system and other reproductive parts. Endoderm forms parts of digestive and respiratory system. For better understanding and visualization, there is a video in YouTube which can be seen.
If we look at a cross-section of an embryo of the frog Xenopus, we can see that at this point it is a ball of cells with a fluid-filled cavity. The cavity is the blastocele, and the embryo is currently in the blastula stage of development. A blastula contains large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The three colors represent the three tissue layers that become defined early in embryogenesis. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells, called bottle cells, move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a few surface cells. The movement of cells into the embryo creates a lip, called the dorsal lip, over which sheets of cells continue to move inside. At the same time, the ectoderm extends around the embryo surface in a process called epiboly. As gastrulation proceeds, a cavity, called the archenteron, forms while the blastocele progressively shrinks. The archenteron is the primitive gut and is completely surrounded by endodermal tissue. The endoderm at the roof of the cavity originated from the outside of the embryo. The cavity is continuous with the outside via the blastopore, which eventually becomes the anus of the animal. As the ectoderm extends around the embryo, another set of bottle cells forms. These cells migrate into the embryo, and other surface cells follow them, creating the ventral lip of the blastopore. By the end of gastrulation, the ectoderm has surrounded the embryo. Endoderm lines the inside, and mesoderm lies between the two. Additionally, the fates of specific regions have become determined. The endoderm gives rise to the digestive and respiratory tracts and associated structures. The mesoderm gives rise to the skeleton, circulatory system, muscles, excretory system, and most of the reproductive system. The ectoderm gives rise to the skin, sense organs, and nervous system.
So this is all about early development of frog and this lecture. Major parts of this lecture are also prepared from the book Developmental Biology by Scott F. Gilbert. Thank you so much.